Hi, I'm Dr. José María Llobet. I'm professor of medicine at the University of Barcelona Hospital Clinic and also at Mount Sinai School of Medicine in New York. I'm going to talk today very shortly about the paper we just published in GAT about inflamed and non-inflamed classes of HCC, a revised immunogenic classification. This is a study that updates and modifies partially the classification we published in gastroenterology in 2017. Very briefly, as you know, HCC is the third cause of cancer-related death. There are more than 850,000 new cases yearly. HCC accounts for 90% of primary liver cancers, and the immune or inflamed phenotypes of HCC have not been fully characterized. It is known that biomarkers predicting response to checkpoint inhibitors alone that achieve that in 15% of the cases, or in combination, particularly atezolizumab plus bevacizumab, that achieve that in 30% of the cases, are poorly defined. Here I'm showing the current frontline and second-line therapies for HCC. In frontline, we have atezo-bev and durvalumab for patients that do not have any contraindication for immunotherapy, sorafenib and lenvatinib for the others. And then in second line, we have regorafenib, cabozantinib, and ramucirumab, but also in the US, nivo-ipi and pembro in second line for patients that progressed sorafenib and lenvatinib as well. So there is a need to understand and properly characterize the inflamed subclass of HCC. We previously reported in Gastroenterology 2017 the immune class of HCC that represents around 25% of the cases. However, after that, we saw that the association of response to checkpoint inhibitors do not totally overlap with this immune class, and also the immune traits of the remaining 75% of the patients were ill characterized. And also that what we define as the immune excluded class that was completely enriched by beta-catenin mutations, actually they might correspond to two subtypes of cancers with beta-catenin mutations. And for this purpose, we analyze a new cohort of patients, around 250 patients, with a full spectrum of next generation sequencing and transcriptomic analysis and immune related analysis, all types of molecular analysis associated with clinical data. And the new findings are these three. The first one is by using this integrative genomic analysis, we have identified the inflamed class that actually expands what formerly was the immune class. The immune class accounted for active and exhausted class, and now we have described the immune-like subclass that enlarges the inflamed class with a new category of patients with a substantial immune infiltrate of CD8 and macrophages, but at the same time that have beta-catenin mutations and low chromosomal aberrations. This class can be identified by a 20 gene signature and is associated with response to checkpoint inhibitors. And also we describe the non-inflamed class at the same time as the intermediate and excluded. Mostly these classes are characterized by a low infiltrate of immune cells, particularly the excluded class that in other tumors is called immune desert class, and a high number of chromosomal aberrations. Thirdly, the third discovery of our study was that we were able to characterize and decipher the potential dual role of beta-catenin mutations in terms of immune response and evasion. So there is a subgroup of patients with immune infiltrate, the tumors have immune infiltrate, but at the same time beta-catenin mutations, whereas others that have completely excluded phenotype with paucity of TILs high chromosomal aberrations, but as well they have beta-catenin mutations. And in this sense, when we analyze beta-catenin mutations, 70% of these beta-catenin mutations are enriched in the excluded class, whereas 20 to 30% are enriched in the immune-like class, and as a result in the inflamed class that represents classically the hot tumors. And therefore are other mechanisms in the microenvironment, particularly interferon signaling, that are able to counterbalance the effect of immune evasion associated with classical beta-catenin activation. In addition, we identify a 20 gene signature that is able to capture the inflamed class, and in an independent cohort of patients that have been treated with checkpoint inhibitors, this inflamed signature was able to differentiate those patients with objective response that you can see here in green, with those patients with no response, either stable disease or progressive disease. Therefore, the conclusions of our study are that the inflamed class that accounts for 35% of the patients include a newly identified immune-like class, whereas the non-inflamed, representing the majority of the patients that are referred to as the cold phenotype, comprise the intermediate and excluded classes. A 20 gene signature in tissue and also a set of proteins in liquid biopsy are able to capture the inflamed hot HCC tumors. And finally, beta-catenin mutations in HCC are both associated to immune exclusion in 70% of the cases, but also with a microenvironment inflamed in 20 to 30% of the cases. And this explains how come now in some reports beta-catenin mutation is associated as well in some instances, in some cases, to immune checkpoint inhibitor response. Overall, our findings can guide the discovery of clinically useful biomarkers of response in the inflamed class or primary resistance among non-inflamed tumors to immunotherapies, and lay the groundwork for the development of new combination-based therapies strategies in HCC. Thank you very much, and hope you enjoy the paper. Thank you.